guys, welcome back to the Xylem IGCSE Biology. Today it's gonna be a new video today. Today we're gonna cover chapter 6, which is plant nutrition. So previously we cover animal nutrition, and then it's all about what humans eat, and some can be related to animals also. So plant nutrition is gonna be really interesting, and then we are gonna recap again with 6.1 types of nutrition. Let me expand this first. Okay, we got 6.2. We will go through a bit of photosynthesis today. Photosynthesis, and then we're gonna go through a bit of the leaf structure also. Leaf structure, which includes the veins, all those, and then we are also going to go through a bit of what is it being used and everything, and then we're gonna have a bit of limiting factors and also rate of photosynthesis, and then some experiments like hydrogen carbonate indicator. It will also be explained a bit. Hydrogen carbonate indicator. Okay, and then indicator, and then we are also going to go through a bit of growing crops. Growing crops and a bit of mineral requirements. Growing crops and a bit of mineral requirements. So we're trying to make this short and easy so that you can get the most out of it. Okay, so we are going to start a bit of something about how types of nutrition that we have learned previously. So we're going to go through this. Okay, so types of nutrition. So remember that a nutrient is a substance which is needed for growth, repair, and metabolism. And then we have two types of nutrition, which are animal and fungi. And fungi is heterotrophs. Green plants are autotrophs. Okay, heterotrophs are organisms that eat other plants and animals. And then autotrophs are basically organisms can produce their own food. Okay, so plants use respiration to break down. Okay, so these are just the types of nutrition, and then next we will enter photosynthesis. So we all know that what is photosynthesis mean is that it's basically green plants needed to make their own food. Okay, we humans eat food by eating, so we all know the equation for photosynthesis is carbon dioxide plus water with light plus chlorophyll to give us the products, which are glucose plus oxygen. Alright, so knowing from here, we can see that there is like carbon dioxide that is being absorbed inside the plant first, and then it produces oxygen out. Okay, so in the chemical formula equation, we are also required to remember this. So in the chemical form of equation, it's going to be 6CO2 plus 6H2O equals to C6H12O6 plus 6O2. This is the balance equation. Okay, so we all know that chlorophyll is the green pigment found in chloroplast. Okay, so a bit of basics on how it works. So water is being absorbed and transported through the xylem from the roots. Okay, secondly, carbon dioxide diffuses into the leaf through the stomata. Okay, the glucose is used to make substances needed by the plant. Okay, so it used in respiration to release energy, ATP. Okay, in the end, oxygen is being diffused out of the leaf through the stomata. Alright, so this should be not too hard to understand. So one more keynote, the light energy is converted into chemical energy in the bonds of holding the atoms in glucose molecules together. Okay, that is all for this. Okay, we will enter the leaf structure, which is a bit of memorization required. Okay, give me a sec on this. Okay, the structure of the leaf. Okay, so we're going to show you a bit of cross section of the leaf. Okay, have a look at this picture. It's a bit too small. Okay, give me a sec there. Okay, so the top of the leaf is the wax cuticle. So whenever you touch the leaf, you feel a waxy kind of feeling. That is the wax cuticle. And then below of the waxy cuticle is called the upper epidermis. So below the upper epidermis will be the mesophyll cell, specifically the palisade mesophyll cell. So the palisade mesophyll cell we're going to talk about more because this is the one that does photosynthesis. And then whatever's in the middle is part of the mesophyll. Okay, and then we have the spongy mesophyll cell where gases exchange are being produced, and then this is at the air spaces. Okay, the stomata is below, right on the lower epidermis. Okay, and chloroplasts are basically the green one on the dot, which is on the leaf itself, the leaf cell, the mesophyll. Okay, so we're going to talk further right now what is about the structure of the leaf inside, and then why is it like so important to remember this, the leaf structure itself? Okay, starting off, we are going to talk about the waxy cuticle. So the waxy cuticle is to prevent cells from drying out. Okay. Secondly, we have the upper epidermis. The upper epidermis is to protect cells inside the leaf. Okay. Third, we have the palisade layer, which photosynthesis happens. Okay. We have the spongy layer. Some photosynthesis happens. Plus a gas exchange. But if you want to ask, since spongy layer has photosynthesis happens, which does the most photosynthesis, it will forever be the palisade mesophyll cell. Okay, why? It does the most because it's closest to the leaf, and then it can produce more chloroplast itself. It has more chloroplast inside the cells than the spongy layer. Okay, we have the stomata or the stoma. So it's basically allowing carbon dioxide exchange. Number 6, we have the vascular bundle, where the xylem and phloem takes into place. Xylem will be the transport of minerals and water. And we got phloem, which is part of the vascular bundle itself, transport sucrose and amino acids. Lastly, we have the lower epidermis. The lower epidermis is to protect again the cells inside the leaf. Same thing. They have a protective layer in up and down. Okay. So phloem transport sucrose, and then xylem transport minerals. Okay. And then the transport structure. I'm gonna draw the transport structure of the vein, which is xylem and the phloem itself. Okay. It's something like this for the leaf, that it has some kind of like shape. Okay, inside it has some few these kind of like egg shape, and then it has one line in the middle. For leaf, xylem is at the top. Okay, this will be the xylem. And the bottom one will be the phloem. Okay, and then we have the stem. The stem will be in circular form, and then inside we have something that looks like, again, like an egg, but more of like an upside down egg. So it will usually looks like this. Okay, the phloem will be at the top, xylem will be at the bottom. Phloem and then xylem at the bottom. Okay, lastly we have the root, which looks like a corona, and then it has, this is much easier. Why? Because it has a star, which is usually shaped like an X. So the X itself is already the xylem, and then what's inside here will be the phloem. Okay, so the star is the xylem, which is X, and then we got the outside layer of the xylem will be the phloem. Okay, so that's all for this one. We're going to delete this, which is much easier, and then done. Alright, next one. We are going to talk about limiting factors and rate. Okay, and then for limiting factors, it's to see limiting factors. Okay, So limiting factors is basically the amount of sunlight the plant is exposed to. Okay, and then it also sees the concentration of carbon dioxide and also the optimum temperature. Okay, so if the graph I'm gonna draw again, which is something like this. Okay, sorry about the drawing also, and then it goes into like this. Light intensity be Li, and then rate of photosynthesis ROP. Okay, for this case, this will be sometimes photosynthesis is limited by the concentration of carbon dioxide. So if there is plenty of light, it cannot do photosynthesis when there is insufficient carbon dioxide. Okay, all right, next one, it's carbon dioxide concentration. Okay, for this case right now, it's the same type of graph, but it's a bit lower this time. Okay, so what is different right now is, okay, this time will be the carbon dioxide concentration, the COC. Again, rate of photosynthesis still is there. Okay, right now it's the carbon dioxide concentration. So what does this mean is that without enough light, a plant cannot photosynthesize very quickly. So even if there is plenty of water and carbon dioxide, increasing the light intensity will boost the speed of photosynthesis. Okay, all right, next one, last one. Okay, we enter close a bit. Right now it's temperature. The graph looks a bit different comparing to the others, that it goes into a curved line. Well, let's just show this because of enzyme. Okay, relate to enzyme immediately from this graph. So this should be temperature, and again ROP, rate of photosynthesis. Okay, at this stage, is if it gets too cold, the rate of photosynthesis will decrease. Enzymes inactive, plants cannot photosynthesize if it gets too hot. Why? Because it denatures at 60 degrees usually. Okay, so this is the limiting factors of plant nutrition. Okay, I'm going to erase this. All right. Okay, the next one, we're going to talk about hydrogen carbonate indicator. Okay, hydrogen carbonate indicator is something like what you call it as the acid and alkali, like that. It's something like that. Okay, but this is a bit different. Okay, so don't get messed up with the colors that you see during the hydrogen carbonate indicator. So the hydrocarbonate indicator. Okay, so it's an indicator used to measure carbon dioxide levels in aquatic system, basically water. Okay, so it is rate in equilibrium in with atmospheric air. Alright, and then it becomes more orange and yellow with increased carbon dioxide levels. Okay, it also changes from red through magenta to deep purple as carbon dioxide is removed. Okay, I'm going to show you a pic for hydrogen carbonate indicator. Okay, so it looks like this. Alright, so the increasing CO2 in indicator will be yellow and atmospheric CO2 level will be here. Decreasing CO2 in indicator will become purple. Okay, so what this means is that, okay, let me put another one. If respiration of CO2 increases, then color will change. Okay, if respiration is higher, become yellow. If respiration equals to photosynthesis, then it will be orange. Number 3, ignore the number. If photosynthesis is higher, then it will be purple. And then if no living cell, there will be no color. Okay. Alright. That is all for hydrogen carbonate indicator. And then next, we are doing the last part, which is the growing crops and minerals. Mineral requirements are also really important for this. Okay, give me a sec for mineral requirements. So, we all know about mineral requirements are so important in this stage for plants itself. Oh my goodness, this is so difficult. Okay, mineral requirements. Okay, mineral requirements are because photosynthesis produces carbohydrate, but plants contain many other types of biological molecules such as proteins, lipids, and nucleic acid, which is DNA. Okay, so as plants do not eat, they need to make the substances themselves. So we have magnesium and nitrogen. Okay, so this means that without salt, these plants cannot photosynthesize, okay, or grow properly. So mineral ions are actively absorbed from the soil. So minerals are absorbed from the soil. Okay, so okay, let's give an example. We have like a bunch of things that what happen if that certain amount of plant that don't have that enough of mineral requirements, they will shrink or what. So we got magnesium. So magnesium is needed for making chlorophyll. Okay, and then if there's a deficiency of that certain magnesium of the leaf, then it turns yellow. Okay, it turns yellow. And then nitrate. Nitrate is to making amino acids, which the deficiency will be stunted growth. Okay, that's all for mineral requirements. That's why mineral requirements are so important for the plant in order to grow. Like us, we need protein. Okay, lastly, we're going to talk about growing crops, okay, as the conclusion of this video, and then in glass houses. A few factors why, because in the glasshouse building, you can maintain the temperature or alter it. Secondly, you can provide shade if necessary with the plant. Okay, and then you can provide sufficient water if needed from the plant. And then lastly, optimum concentration of carbon dioxide. Okay, that's all for glass houses. So these are the important things for 6.1, 6.2, 6.3, 6.4, 6.5, and 6.6. If anything I left out from this video, I can always do an extensive one to in order to make sure that you also understand. If you don't know, drop your questions into the chat in the comments, and then I would really answer all of them for you. So thanks for watching this video, and again, it's a long one. Again, I wish that this is easily understand to you, and then hope you guys do well. And then the next video will always be something interesting related also, and then see you guys again. Bye bye.